Thank you for your interest in this presentation. I have frequently seen in the pharmaceutical industry in North America that the passivation process meets a standard and passes a peroxide test. These two features underpin the validation process. However, demonstrating cleanliness required by the industry nowadays will require a deeper understanding of the standard and a more sensitive testing than the peroxide test. This presentation is divided into three parts. In the introduction, you will understand the problem. I will explain what passivation and validation is, how an interpretation of the current regulations supports the need for passivation of stainless steel. Using a risk evaluation, my 7 years experience, and FDA reports, I will show you how a poor passivation process jeopardizes the production quality and means liability. You will see some examples of contaminants frequently found on new equipment that may not be removed if the interpretation of the standard is limited or the procedure is not the most suitable one. There are many tests to detect the passive layer. Each one has its advantages and weaknesses. By passivation modalities, I mean if passivation is achieved by recirculating chemical, dipping the metal, or use other way to put in contact the metal and the chemicals. Besides caustic and citric acid, many more chemicals are used to clean and passivate. I will mention many of them and their use. In drafting the document section, you can find a very comprehensive list of information to include in a passivation validation package. Finally, I will talk about other stages of the cleaning process associated with the passivation, especially the sanitization and the rugging. The passive layer is invisible. Stainless steel can be passivated or only cleaned. We will see no difference. And its performance in the pharmaceutical industry does not represent an immediately catastrophic risk. With the modern analytic techniques, we can detect the passive layer, prove that the stainless steel is passivated, and we can measure it and see its composition. Usually, the passivation and related documentation in the construction or commissioning of a new piece of equipment in the pharmaceutical industry is an oversight. After all, the passivation cost and execution time represent only a small fraction of the total project. To make the matter worse, the consequences of an elementary passivation are not immediate. Corrosion or contamination resulting from an elementary passivation practice will be seen after some or many years of the initial startup. This presentation helps you to understand the pharmaceutical industry's regulation, standards, passivation, and documentation practices. Passivation of stainless steel is to clean its surface with an acid to remove free iron that is not bonded to the alloy and creates a chromium-rich surface. In this animation, we can see how the passivating chemicals remove iron and left a rich chromium surface. In these pictures, we can see the different types of rust or iron oxide, products of the reaction between stainless steel and, for example, on the left with steam. The picture in the middle is with water, and on the right with saline solutions. But there are other substances like sanitizers that also can react with the stainless steel. The FDA or other regulatory agencies requires that the process must be under control. The written evidence that a process is under control is the validation. The FDA defines validation as establishing document evidence that a specific process will consistently produce a product meeting its predetermined specifications and quality attributes with a high degree of assurance. The passivation validation may be included in the installation qualification if we are talking about something new. It's part of the process qualification because we need to demonstrate that there is no interaction between the stainless steel and the raw materials or products that we manufacture. In the preventive maintenance procedures, because the passive layer needs to be regenerated from time to time, and the change of control if we need to modify our procedures related to passivation. The good manufacturing practices, or GMPs, are like an umbrella that covers all the aspects of drug manufacturing. They tell us what to do, but they are not specific enough to describe how to do it. The GMPs do not mention that equipment should be made of stainless steel, and for this reason, they will not mention the need of passivation. On the other hand, it is a general practice and common understanding that all the stainless steel in contact with a product or raw material to manufacture drugs must be passivated. This practice is a current GMP. The Code of Federal Regulations requires that the surface in contact with processed materials should not be reactive. If stainless steel is not passivated, it will react with materials. In Canada, the Food and Drug Act requires that the drugs are manufactured in sanitary conditions, with the absence of a passive layer in a water loop, for example, or other equipment and systems. The stainless steel will generate ROS. This increases the chances of biofilm formation. I will use a risk-based approach to mitigate the consequences of corrosion and product contamination related to passivation. To determine the probability of occurrence, I will use my 7 years of experience and the FDA 483 observation reports. The methodology starts with identifying risks, such as particles and other construction debris going into the product. Then, using this table, we evaluate the probability of occurrence. I have to accept that I'm still shocked by the fact that we can find construction debris in medication, and it happens more frequently than I used to believe. I have seen this specific case every 1 to 5 years. It is for this reason that I will give it a score of 3. Based on this chart, I classify the consequences as critical with a score of 4. The consequences depend on the product and the type of contamination. The risk is not the same if you manufacture a topic medicine with dust particles, or if you manufacture an oral medication with metal shavings. These charts guide the classification of impacts. A critical consequence means that the authority may conclude a serious non-compliance or request product recall. The liability and direct or indirect repair costs between $100,000 and $1 million. We combine the scores of probability and impact in this table to get a risk rating. The green zone means an acceptable risk. The orange zone means a manageable risk. And the red area is an unacceptable risk that need to be eliminated or reduced. For our exercise, a possible risk with a critical consequences give us a risk rating of 12 in a red area. This means that we need to implement action to mitigate the risk. In this slide, You can see more detailed information about the risk rating by colors. Some of the controls that we can implement to reduce the risk of construction debris in our product are construction procedures. Control turbulence and time when flushing pipes during pre-operational cleaning. Use of filters or screens during pre-operational cleaning. Install metal detectors. We organize the information in this table. We additionally mention that construction debris also can harm equipment, as well as the customer, the product, and the company's reputation. We assess the risk again with our controls, so the probability that construction debris goes into the product is very rare. The consequences are negligible, and the residual risk is acceptable. Here we can see the evaluation of microbiological contamination associated with corrosion or biofilm in critical water and steam systems. The risk is more frequent in cold systems. However, I have seen bacteria growing in clean steam systems. The classical culture approaches of water testing, such as pour plates, spread plates, membrane filtration, and the most probable number, do not always detect biofilms. If we control the microbiological quality of water with a method which is not sensitive to biofilms, we have the risk of regular undetected contamination. In other words, sometimes we have positives, others false negatives. Some of the controls that prevent biofilm formation are improved sanitization procedures that include every component in a water system and the rotation of sanitizers. Keep the surface of stainless steel free of corrosion and scales that protects and facilitates the biofilm formation. And, of course, let a microbiologist plan in advance for biofilm detection. The last risk that I will evaluate is corrosion. Sometimes stainless steel structures, such as stairs and platforms, are cleaned with corrosive sanitizing agents. Chlorine compounds are the most common ones. A corrosive environment, such as a hydrochloric acid fumes, also leads to corrosion in stainless steel. Dusty iron oxide in autoclaves and lyophilizers can contaminate the product and the packaging. Some of the controls are the use of non-metallic materials, the use of non-corrosive sanitizers, adequate construction procedures, and better passivation practices.